So just to recap on a couple of terms that might be useful. So DNA, this is the information needed to make and maintain us. And our DNA is made up of these building blocks of chemical bases, which we refer to in this cartoon with the letters A, C's, T's and G's. So you can almost think of these as the alphabet of life. We all originally came from the same evolutionary thing. So the A, C's, T's and G's that are in you are exactly the same as the ones you'd find in a wheat plant, for example. When you hear reference to genetic sequence or genomic sequence, it means we now have the technology which allows us to read the order in which these letters appear throughout our genome. And something like the wheat genome will contain 17 billion bases or letters. And genetic variation is, if we were to compare the genome sequence between species, we'll see that there's a different order of letters, and that's genetic variation. And there was a really interesting study where 400 genomes of tomatoes were compared, and they found that some had 33,000 genes and some had 36,000 genes. So you can take away 3,000 genes and it's still a tomato. But most sort of genetic variation is much more subtle changes to that. So it can just be deletion of a few bases or just the changeover in that order in which bases occur. So that genetic variation can be a result of natural mutations. It's the basis of all evolution. And this is what plant breeders have been looking at for centuries. So they look for mutations or changes in the order of letter that gave rise to different traits. So for example, something like these different vegetable types, they're all the same species, but different traits that have been selected for over time have given us bigger leaves to give us kale, or denser flower bud formation to give us things like broccoli. And a plant breeder can only select from the genetic variation that's available to them. So it's very much a case of sort of hunting and looking for it. And so in the 1930s, a method called mutation breeding was introduced as a way of speeding up the occurrence of these mutations or genetic variation. So what's happening at the molecular level is you can lose sections of DNA, or you can create a break in the DNA, and as the plant attempts to repair itself, it may delete the odd base or recruit the wrong letter. And so you've introduced a point mutation. And then a breeder would look to see whether new desirable traits were found, and then he would breed those into his breeding lines. The problem with mutation breeding is you're introducing hundreds of thousands of random mutations into your lines, and there's no control of where they would be found. Whereas technologies that we have now, so we have access to all this genomic information. So if you think that plant breeding till now has kind of gone on without any knowledge of DNA and genetic sequence. So we now have access to all that information. And we have technologies, so genetic technologies, things like GM or gene editing, which now allows us to go in and make very specific changes to an organism's DNA. And just to sort of perhaps relate bases to genes, a given gene can be anywhere between a few hundred bases long, or it could be a couple of million bases long. So genetic technologies. So gene editing is an example of new genetic technologies. There are a number of different gene editing tools out there, but one called CRISPR-Cas is probably the most popular. That stands for clustered regularly interspersed palindromic repeats, which doesn't help explain the technology, but it was first identified in bacteria. And it was first used to edit genes in plants in 2013, and we've been routinely using it now at the John Innes Centre to edit the genomes of brassica crops, wheat crops, barley crops, potato and tomatoes. And so you can think of this as a tool which allows us to make cuts to the DNA at very precise locations. So we now have genomic sequence for most species that we work with. So we can now direct where we make breaks in the DNA. So we can delete sections of DNA, or we can make a break to the DNA. We hope that the plant attempts to repair that break, and in doing so, it could delete or insert the wrong base. So we're not adding anything new, we're just making deletions or cuts to the DNA. And we would refer to these as gene knockouts because these small edits are often enough to stop that particular gene from functioning. And these would be regulated as precision-bred organisms under the new Genetic Technologies Precision Breeding Act, because the endpoint could have been achieved through traditional breeding approaches, but we've just gone there in a much more direct route. Now, we can also use this technology to open up the genome at a precise location in order to insert or replace genetic sequence, and we would refer to these as knock-ins. These would continue to be regulated as GMOs if the gene was from another species. So if we were to take a disease resistance gene from a wheat plant and put it into tomato, they would count as transgenic. So they're going from one species to another, and they would need an additional regulatory oversight. But if we were to take a gene from, say, a potato plant and reintroduce it into a potato plant, this would be referred to as cisgenic, because the endpoint could have been achieved through traditional breeding approaches. And in the picture we've got below, this is an example of blight-resistant potatoes. So you can see the nice green plants. So these contain 3 genes from wild potato relatives that have been introduced into commercial varieties in order to withstand late potato blight. So this is a really nice example of where we can use biological solutions to tackle these sorts of diseases rather than rely on fungicides. But I'm going to focus more on the gene knockouts because this is the majority of crops that are coming to market or nearing commercialisation will be where we've made deletions or small edits to the plant's existing DNA and where we've not introduced new genetic sequence. So this is a worked example of where we've used gene editing to create a gene knockout in order to identify or confirm that that particular sequence of DNA controls that particular trait. So what I'm showing you here is a field of oilseed rape. Now our farmers face some great challenges in even getting to this stage, but what a farmer doesn't want to do is lose some of the seed at the end of that as a result of premature pod shatter. So can we use gene editing to edit the genes responsible for pod formation in order to stop those pods shattering prematurely in the field and losing yield? So in this rather frightening looking slide, what I'm showing you is an example of genomic sequence. If I was to show you the entire genome sequence for brassica, I would need to share with you several million screenshots like this. So vast amounts of genetic information that we have available to us. And by comparing genomes from different species where we have knowledge of which genes control which traits, we can predict which gene sequence or which sequence of DNA we believe controls that trait in this crop. So that becomes our candidate gene. This is all done on a computer at this stage. So we find the sequence that we believe controls that trait. We would then look to identify a 20 base sequence that is unique to just your gene and nowhere else within the genome. And then we'd design a molecular guide which we'd introduce into the cell, which would hunt out and search for that genetic sequence. And you can think of this a bit like a Google search engine. And once it finds that sequence, it locks on. And then we introduce a nuclease or a Cas protein, which cuts the DNA at that location. So what we then hope is that the plant will attempt to repair itself, and in doing so will introduce an error. And this is what it looks like in reality. So here you can see our original genetic sequence at the top, where we've designed our guide, and the circle shows where we've made that break. And you can see that the sequence has changed in the edited line, so a C has been replaced with a T, and we've lost a G base. Now the image that I'm showing you is a cross-section of a fruit pod, and where the green arrow is showing is where that valve margin forms, so that's at the point at which those pods would break open. And in the edited plant, you can see that valve margin no longer forms, and so those pods would need to be broken mechanically in order to release the seed. Now for us, this wasn't a commercial endpoint for this particular project. It was really for us to use these technologies to confirm this is what that gene does. So because although we've got access to this genomic sequence, we don't know what everything does in that. So these technologies are really powerful research tools for us to help us understand what genes control which traits.